when you see what happens with potassium if it is in excess outside the cells, which can happen. Yeah. What else?
Kidneys.
Kidneys. Yeah. Where are they? Where are they located?
Retroperitoneum.
Right back here. Did you know kidney punches are illegal in boxing? You know why?
Oh.
Yeah, kidney punches are illegal. And the reason they're illegal is because your kidneys have very little protection. And they are loaded with blood. 20 to 25% of all your cardiac output is coursing your kidneys at any given time. So if you get a kidney punch, it does a lot of damage to the kidney. And then you may lose the kidney. Which fortunately, we have 2 of them. So people can lose a kidney and still be able to function. Anything else?
UTIs.
Oh, yeah. Especially, yeah, urinary tract infections for the females because,
You have all that bacteria.
yep, it's right. We talked about it for when we did the urinalysis. Yep, you can have bacteria traveling back up through the external urethral sphincter, up the urethra, and into the bladder, and then from the bladder up to the kidney.
Sorry, quick question. Is the urinalysis stuff that we did in lab on the lab test? Okay, that's what I thought. It's just
That's why I wanted you to do those case studies,
okay.
just so you have some sense about looking at the results and figuring out what's what. Are those normal results, abnormal results? What might those results lead to? Yeah. All right, so yes, we have 2 kidneys, usually, unless, of course, you've lost a kidney. Or you have something called a horseshoe kidney. Believe it or not, 1 out of every 500 or so people has a horseshoe-shaped kidney. Which means that potentially, you probably know 500 people, you know, extended, you've met somewhere along the line, work, put all your contacts together. You may know somebody who has a horseshoe-shaped kidney, you would never even know it.
One of us in this room might
Maybe,
have
could be. You would never even know it unless you have problems. You might have 2 kidneys, but one of them is in the correct position, one down here in the pelvis, so it's known as pelvic kidney. Or you might have your kidneys placed as what we would refer to as normal. Yeah, so we have 2 kidneys. They are very large. They're like the size of a bar of soap. My fist, maybe that's a big bar of soap, but a bar of soap, somewhere in the Dove range, maybe a little bit larger than that. So it depends on your size overall. 2 of them, you might see that the right kidney is a little lower than the left kidney. What do you suppose is causing that?
Right longest, 3 livers.
Liver.
Oh,
Yep, the liver. The liver is located right there, so it pushes it down a little bit. Yes, they both produce urine as a process of filtering the blood. And a way of filtering that blood is to remove the waste products. So, I'll go on with this slide and then I'll go on with my story. So we have 2 kidneys, we have 2 ureters. The ureters are the tubes that carry the urine from the kidney to the bladder. We have 1 urinary bladder, and I always need to be reminded to say urinary bladder because we also have a gallbladder. So I know almost everybody thinks of bladder as the urinary bladder because we rarely talk about the gallbladder because, you know, what's the gallbladder do? It just makes bile. I mean, it doesn't make bile, it stores bile. Yeah.
Gallbladder is like one word though.
And that's another thing. Gallbladder is 1 word, and this is urinary bladder. Yeah.
So does gall
I'm not going to get upset with you if you just say bladder.
Okay. So does gall mean bile?
Good question. I haven't looked that up. The urinary bladder is where we store the urine, and then when the urinary bladder gets too full and the pressure is excessive, and your parasympathetic system says your bladder is stretching, go empty it, then you do. And that is referred to as micturition. That is the official term for urination, or peeing, as we all call it. The urethra is from the urinary bladder to the outside, and interestingly enough, this is one of those systems where everything is actually exterior, because we have from the urethra up to the bladder, up the ureters, and into the kidneys, all exposed to the outside, ultimately. Just like the respiratory system, digestive system, reproductive system, all actually exterior, which is kind of weird, yeah.
Do you know if moisture kidneys are more efficient? Because they have more larger surface area?
Don't know about that, hadn't heard that,
That's why I was wondering if it was like a stronger kidney.
but I do know that they can cut them and use them as transplants. If somebody were to accidentally die and they had a horseshoe kidney, they could actually cut those up and use them as transplanted organs.
Wow.
Yeah. So here's what the urinary system is famous for. Obviously, it is for excretion, for getting rid of waste products, metabolic waste in terms of urea, creatinine, uric acid, ammonia, if the ammonia hasn't been converted to urea. The kidneys are very good at doing that. And as you see here, obviously we remember that the respiratory system is responsible for getting rid of excess CO2. The digestive system is responsible for getting rid of all the waste from the nutrients from the food that you've eaten. And the integumentary system, what's the integumentary system's responsibility in terms of excretion?
Sweat.
Sweat, yes.
What's the difference between excretion and elimination?
Elimination and excretion. Excretion is through glands and tubes and
Oh,
pores
okay.
and tubes.
Okay.
And I think elimination might be considered a more generalized term.
Okay. I was just looking at the, like, it separated the three primary functions. So I was just wondering why excretion and elimination were separated into separate.
Yeah. So elimination,
Is micturition.
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 And then the kidneys also have all of these very important jobs in terms of maintaining our homeostasis. When you see this, you kind of think about water, because our blood volume and our blood pressure is based on how much water is in the blood. When we're dehydrated, our blood pressure can drop because we have lower blood volume. Concentration of plasma ions, this osmolarity, is the moving around of sodium and potassium and calcium and chloride and a number of other ions that are coursing through the system in our kidneys. Extracellular fluid pH. Why is that extracellular, not intracellular?
Does it have any outside of this now?
Of course. It's where we can control it, right? We can't control it inside. Unless we control the outside, we can control the inside only through the outside. So, yeah. Nutrient conservation. Glucose, biggie. Plenty of glucose in your blood. You want to keep it in the blood, but as I describe in a moment, you'll see that glucose gets bounced back and forth. And glucose can actually act osmotically and draw water with it, which is one of the problems for diabetics. Making red blood cells, erythropoiesis, because erythropoietin is produced in the kidneys. Remember that from 232? Calcitriol. Calcitriol is the result of calcidiol, and calcidiol from cholecalciferol, vitamin D, and the retention of calcium ions to maintain that we have plenty of calcium in the circulation. Gluconeogenesis, what's this mean? Gluconeogenesis.
Creation of glucose.
Making new glucose, yes. So if you are in an extremely starvation state, your kidneys and even your liver can get involved in helping you create new red cells. Don't ask me how, I haven't investigated. Detoxification of drugs, removal of hormones, all that back up to getting rid of waste, right? So here's what happens in the kidneys. Imagine you're moving and you have stuff, like we all do. You have stuff that you want to take with you. You have stuff that you don't necessarily want to take with you. There's stuff that you can't take with you. So you put all the stuff in boxes and you take all that stuff and you put it outside. This is what the kidneys do. The kidneys take literally everything out of your blood that can be removed from the blood through filtration, and all of that material is sent to tubules. The things that the kidneys cannot remove from the blood include red cells, white cells, platelets, and large proteins. Those would be like your refrigerator, your dishwasher, your sink, your any large appliance, things that you can't move out of your house or your apartment. They stay in the house or apartment, which now is your blood. But everything else is pulled out of the blood. Glucose, calcium, magnesium, chloride, potassium, urea. Everything and anything that can be pulled out of the blood will be taken out of the blood at a little structure called a glomerulus. It's the functional unit, part of the functional unit of the kidney. And then all of that stuff that's been pulled out of the blood is put into a fluid called filtrate or tubular fluid. And slowly, as that fluid is coursing through the kidney tubules, materials are brought back into the blood. Glucose is retained, calcium is retained, sodium and chloride are retained, potassium is retained, urea is retained, and then urea is shipped back out again, and then back in again. But the point is here that you are moving, you have boxes out there in the yard, and you go, oh, I don't need this box, I'm gonna get rid of that. That would be the urea. Oh, I need this box, I'm gonna take it back into the apartment until I'm ready to load the truck. That would be your glucose. That would be your calcium. Can you see how the kidney's working? So it's filtering out all the things that we don't need, and all the things that we do need, we keep and put back into the blood through this process called reabsorption. Then there's some things like, oh, last minute you got this box, oh my God, you decide you definitely don't want it, you're gonna get it out at the last minute. This would be referred to as secretion. So reabsorption happens where we pull everything back into the blood that we need, glucose, calcium, sodium, chloride, etc. All those electrolytes pulled back into the blood. The urea actually is used as a little sponge, which we retain water. Oh yeah, I forgot to mention water. Water is going to follow all those electrolytes because they're all osmotically active, especially sodium and chloride. And then we have some hormones that help manage this whole moving process too. So that's the basis of how the kidneys work. Filter everything out, and then retain what we need, get rid of the rest. Maintaining homeostasis is a biggie for the kidney. Biggie. Water conservation. Conservation and retention of all of the electrolytes that are essential. And this is just describing to you how blood calcium is moved through the system. And in this case, we have our blood calcium levels that are, depending on where they are, 10 milligrams per 100 milliliters, a significant amount of calcium in the blood. If your blood calcium levels fall, then you might recall this from 232, you have these glands, the parathyroid glands, that produce parathyroid hormone. And parathyroid hormone stimulates your kidneys to take up more calcium. Remember that part? And the reason we need calcium. What's the reason we need calcium?
Muscle contraction.
Muscle contraction, right? What else?
Bone density.
Bone density, what else?
Nerve.
Nerve innervation, right? Yeah, you need calcium for all those things to happen. So we take up calcium in the kidneys, and then our blood calcium levels rise. Kidneys also produce this calcitriol, again increasing take-up of calcium from your intestines, right? To take the excess calcium that you produce through the diet and increase that uptake in your intestines. Blood calcium levels rise again. What if your calcium levels are high? Your rising calcium levels. Now we're not doing parathyroid, we're looking at the thyroid gland, and the thyroid gland produces calcitonin. Remember all that?
Mm-hmm.
Are adults influenced by calcitonin? No, right? It's children. Because children have a great need for building bone. We adults, we depend on maintaining that calcium in our bone, not adding more necessarily. Calcitonin tells the kidneys you don't need to keep as much calcium. You can let some calcium slip through. One of the boxes that you're not going to take with you when you move. Calcium out. You got calcium deposition in your bones, good, keep those bones strong. Your blood levels of calcium decline, and boom, you're back to your normal levels. So one of the many mechanisms by which the kidneys are helping us maintain our calcium and our blood electrolyte levels. And we talked about this when we were doing our urinalysis last week. On our urinalysis last week, we talked about how the kidneys are responsible for removing these nitrogenous waste products, and those are including urea. We also talked about urea in terms of how amino acids get deaminated, and in that process, remember we were talking about, right? Yes, changing protein, what we do with proteins. And that urea is a product of this ammonia that is produced from the deaminization of those amino acids. So ammonia is toxic, got to convert ammonia by adding CO2 to it, making it urea, and then that urea, you're going to find out, is really fascinating because we actually depend on urea to maintain our water conservation abilities within the medulla of the kidney. More on that as we get through this stuff. Remember, creatinine is a byproduct of creatine phosphate, and creatine phosphate is a byproduct of having excess ATP produced. So if you have a lot of ATP, cool, you can take some of that phosphate off the ATP and make creatine phosphate, and then you have extra phosphate when you need it. You can make more ATP via ADP. And we have uric acid. Uric acid is a byproduct of the metabolism of DNA and RNA. And why are we metabolizing DNA and RNA?
Because we eat cells.
Because we eat cells. And all cells have DNA and RNA in them. Yeah, so we got to break them down and reuse the parts. Really cool. All right, what else? Some of the things that can happen about our kidneys, happen to our kidneys. Diabetes mellitus. What's diabetes mellitus?
No.
You had a question?
Yeah.
Okay.
Going back a little bit, sorry. One of the questions on our lab exam this week was the smell of ammonia in urine.
You mean the urinalysis lab?
Urinalysis, yeah.
Yeah.
I know that it can be from infection.
Mm-hmm.
Why? I see here that it's combined with CO2 to form the urea. So does that mean we don't have enough CO2 to combine with ammonia?
Now think about what's the ammonia from? Bacteria.
Right.
So ammonia is indicative of bacterial infection.
But the ammonia that's being turned
Produced
into
here is going
urea,
to be turned into urea.
we're not smelling that.
Exactly.
That one's not coming out. It's a different ammonia that bacteria is
Yes.
making that you're smelling in the
okay.
urine.
Yeah.
Okay,
Yeah.
that makes sense.
This ammonia is the ammonia that we actually manage.
Okay, so is it?
But if we have kidney failure, then we could have that
Oh, okay.
without a bacterial infection. Yeah.
Okay, thank you.
But if you're normal, right, you don't have bacterial infection, then you smell ammonia, something's wrong.
Right, that makes sense.
Or if you smell ammonia, you get urine analysis done. See if you have bacterial infection.
Right.
Culture. Good question, really good question. All right, so what's diabetes mellitus?
Inability to make insulin.
It's the inability to make insulin, or the lack of receptors for insulin. And what's the result of diabetes mellitus?
Inability to process glucose.
Can't process glucose. Where's that glucose found?
The urine.
Maybe.
The blood.
Prior than getting to the urine.
The blood.
It's in the blood. Your glucose is in the blood. Glucose is osmotically active. Osmotically active. See, I'm really tired. Osmotically active, which means that it's going to pull water. It's going to cause high blood pressure, hypertension, right? Yeah.
Oh, duh. I don't know that.
Yeah. And then this hypertension is really problematic because the little capillaries in the glomerulus, this little structure that is doing the filtering, these capillaries are very delicate, as all capillaries are. And so you can imagine if you have really high pressure in that blood coming into the glomerulus, it can blow out the capillaries. And then you get nephritis, or glomerulonephritis. That's problematic, because now the glomeruli aren't working for you, and they're not filtering your blood correctly, and it all kind of backs up on you. Polycystic kidney disease, can you imagine? Cysts growing in your kidneys? And your kidneys literally are a cluster of blood vessels and tubes. And if you have cysts growing in there, then you have no filtration, blood vessels, polycystic disease, problematic. Glomerulonephritis, right, this inflammation or infection of the glomeruli can go right back to any one of these other concerns. So these are some of the kidney problems that can result in kidney failure. This right here you want to remember. Blood urea nitrogen, or your BUN. The BUN is a test that is used to measure the nitrogenous waste products, i.e. urea, in the blood. And it's a pretty stable number, as you can see here. Pretty stable number. And if it is elevated, it's saying that the kidneys aren't capable of removing that urea, so the urea levels rise. Every one of you who's had blood work done has had a BUN, looking to see that your kidneys are still functioning properly. If it is elevated, this blood urea nitrogen, it says that you have a condition known as azotemia. And as you see here, that's a name for a laboratory finding. And the laboratory finding is because you see elevated urea. And if your urea is continuing to elevate, just like everything else, it's going to back up from the kidneys into the blood and causes a condition called uremia. And if uremia gets out of hand, you can see it could pretty much be the death of you.
Fun times.
Because,
Fun times.
Fun times, yes. Any of these nitrogenous waste accumulations can lead to uremia. But uremia from that blood urea nitrogen, from urea, which is a byproduct of your protein metabolism. Think about that high protein diet. There you go. Another reason. Yeah.
So I remember from medical terminology that the nephron, I don't know how you say that, but was
Nephron.
like the functional unit of the kidney. So what's the difference between the glomerulus and the nephron?
The glomerulus is part of the nephron,
Okay.
and we're going to talk about it. We're heading that way.
Okay.
All the components.
Okay.
Yeah. So the kidneys themselves, obviously very important organs. Again, we have two of them. We're fortunate. We can lose one and still be able to function. We like to keep them both. As it says, not so big. They have some unique characteristics. Kind of like to look at the big picture here. When it talks about the hilum, it is the entry and exit for the major blood vessels, and the pelvis and ureter, coming into and out of this indented region in the kidney. Everybody knows what a kidney bean looks like.
Mm-hmm.
Kidneys are the same shape as kidney bean. Which came first, the kidney bean or the kidney? My guess is the kidney. So the hilum is where everything enters and leaves the kidney. It's that indented region. The kidney is considered retroperitoneal. What does that mean? Right? The peritoneum is sitting in front of it, so
Yeah.
the peritoneum is anterior to the kidneys. And the kidneys have several connective tissue structures wrapped around them that we're getting to in a moment. The outermost connective tissue structure that surrounds the kidney is called the renal fascia. And just deep to the renal fascia is the perirenal fat capsule. And just deep to the perirenal fat capsule is the fibrous capsule that is intimate to the surface of the kidney. The renal fascia, nice way to hold things in place, right? The fat capsule, good for insulation, good for shock absorption, something that helps prevent damage to the kidneys. And of course, a little bit of fat stores. This I find really interesting. Imagine that you've lost so much weight. You've burned through all your fat stores, all your adipose tissue. Now you have nothing holding your kidneys in place. You can develop a condition called nephrotosis. nephrotosis, where your kidneys literally are floating. And when that happens, you can have twisting of tubes, like your ureters, like your blood vessels. Not good. Don't want that. So maintain your fat stores around the kidneys. The fibrous capsule is that connective tissue that literally is just intimate to the kidney itself. And this is a, obviously, cross-section that they are trying to depict the different connective tissue structures that are surrounding the kidney relative to the other structures in the body. Here's the renal fascia, the green line, the perirenal fat capsule, the yellow line, and the fibrous capsule intimate to the surface of the kidney. Here is your hilum, where your renal artery enters, the renal vein leaves, nerves, lymphatics, everything coming going in that little indent, that divot in the medial aspect of the kidney. Another way of looking at it. Here's our real kidney, here's our diagram of a kidney. And so some other things for us to pay attention to is this term parenchyma. The parenchyma is referring to this portion of the kidney where all those functional units are located. This is the parenchyma. All the functional units are located there. And by functional units, I'm referring to the cortex, where the nephrons are located, the pyramids, where the collecting ducts and the papillary ducts are located, and the columns, which separate the pyramids. Another interesting thing, and I wish we had a good way of seeing it in an image, there's a space called the sinus, the renal sinus, where literally the renal artery, renal vein are located and some fat surrounding them. So there's a little space right here, you can see a bit of a blood vessel. There's some spacing under here. The renal sinus, as you can see, includes the renal pelvis, the renal artery, and renal vein. When we get to lab and we're looking at the kidneys, we're going to dissect kidneys. We'll try and sort out the difference between the hilum, where everything enters and leaves, and the sinus, which is literally a space. It's hard to describe without being able to see it.
Is there any liquids in the sinus of
Nope.
the kidney?
Nope, no liquids. Just things passing through it. Just a space. Open space.
Oh, I see that now, sorry.
Yeah, that's all right. All right, the cortex is that outer portion. Right along here. Cortex, remember cortex always outside, the cork of the tree. That's where cortex came from. So cortex is always going to be on the outside. And the cortex is where we have the nephrons. And contained in the nephron is a glomerulus and several tubules. There's a proximal convoluted tubule, there's a tubular loop or loop of Henle, and then there's a distal convoluted tubule, and then we hook those up to collecting ducts, and the collecting ducts merge to papillary ducts. Cortex is where we find the nephrons. The medulla is where the papillary ducts all converge and dump urine into structures called calyces. We have major calyces and we have minor calyces. The major calyces receive urine from one pyramid, and then the minors, the minors collect the urine from the pyramid, and then the minors converge to form major calyces. And the major calyces dump that urine into the pyramid, into the pelvis. Minor calyces, major calyces, renal pelvises are all going to be, as you see here, located in the renal sinus. The real thing, this is a pyramid. This would be referred to as a lobe, pyramid and column. Here's a lobe, a pyramid and columns. One pyramid, another pyramid, so this would be another lobe here, pyramid, column, column, et cetera, et cetera, et cetera. Cortex, where the nephrons are located. Medulla, where the collecting ducts, papillary ducts enter into minor calyxes. The minors converge to form major. These guys converge to form a major, and then all the majors dump into the pelvis, the renal pelvis, this region right here. And the renal pelvis then delivers that urine to the ureter, and the ureter to the bladder. And I've said this several times, a lot of blood is coursing through the kidneys at any given time. Cardiac output, 20 to 25%. And the reason for this is because we have a lot of blood to filter. And it's happening as you're sitting there. Your kidneys are working for you, filtering that blood, producing urine. And depending on how much water you've drunk, you're going to make more urine, or you're going to make more concentrated urine. Because if you don't have the water in the system, the kidney's job is to make sure you keep water in the circulatory system, because your blood pressure is very important to maintain a certain blood pressure. We're going to filter that blood, get rid of the waste, keep everything else back into the blood. I'm going to use our kidneys, as this says, calcitriol, right, renin, lots of things that happen, erythropoiesis for maintaining red blood cell counts, etc., oxygen, right? All of those things are tied together. All right, so the blood is a very important aspect of how all this works. And this is what we have in terms of a blood supply and where that blood goes. So we have a renal artery that's bringing freshly oxygenated blood to the kidneys. Because all the kidney cells need oxygen. But that blood is also carrying everything else. Hormones, toxins, waste, good stuff, bad stuff. That renal artery is going to branch into what are known as segmental arteries. The segmental arteries are going to branch into interlobar arteries. Because this is a lobe, this is a lobe, this is a lobe. Segmental interlobar. Segmental interlobar. Segmental interlobar. Those interlobar arteries branch to what we call arcuate arteries. And I like to think of them as forming arches. Arcuate arteries. The arcuate arteries further branch into what are known as cortical radiant arteries, up here. You could also call them interlobular. I personally like cortical radiant because it tells me where they are, the cortex, and it tells me how they radiate off of that cortical radiant. And the way they radiate off is towards something called an afferent arteriole. The afferent arteriole is delivering blood, this freshly oxygenated blood, to the glomerulus, the little cluster of capillaries. The glomerulus sends the blood to afferent arteriole. The afferent arteriole sends that freshly filtered blood to the peritubular capillaries, or the vasoracta, depending on which nephron we're talking about. These peritubular capillaries become cortical radiant veins. The cortical radiant veins to interlobar veins to the renal vein. What's missing?
Segment.
No segmental veins.
Yeah.
Don't ask me why. It worked. So that's our blood supply, and you want to know this. The aorta, obviously, all blood coming from the heart via the aorta. Renal artery to segmental arteries. Segmental arteries to interlobar arteries. Interlobar arteries to arcuate arteries. Arcuate, forming little arches. The interlobar arteries to the cortical radiant arteries. Cortical radiant arteries to the glomerulus. Glomerulus to the afferent arteriole to the peritubular capillaries, cortical radiant veins, arcuate veins, interlobar veins, renal veins, back to the inferior vena cava, back to the heart. Lungs first, then heart. Heart, lungs, heart. Now, mentioned this a couple times. The nephron. This is known correctly as the functional unit of the kidney. This is where the magic happens. Quite a few of them, 1 million or so per kidney. 50 millimeters, that's quite long. But of course they're packed into the kidney, because remember the kidney is only this big. And so the tubes aren't straight, they're very tortuous, they're really twisted. So the nephron contains this glomerulus, which is a little capillary bed inside a capsule. It contains the proximal convoluted tubule, the descending loop, the loop of Henle, or nephron loop, ascending loop, limb, distal convoluted tubule, and then our collecting duct. This says we have a renal corpuscle and the tubules. And the renal corpuscle is the glomerulus plus the capsule surrounding it. Blood in through the afferent arteriole, through the glomerulus. Filter happens here. Everything that you want to move out of the blood, that can be moved out of the blood, is pulled out of the blood via hydrostatic blood pressure and put into this capsule. Anything that can't get out of the blood stays in the blood. What are the things that cannot get pulled out of the blood because they're too big?
Large protein.
Red cells,
White cells.
white cells,
Large protein.
large proteins, and
Plasma
platelets.
or platelets?
Yeah, plasma's liquid, so water gets pulled out.
That's what I meant.
Water follows everything. Water follows everything. But you were on the right track. So we have the renal corpuscle here, including the glomerulus and the capsule. We have our proximal convoluted tubule, down the descending limb of the loop, up through the loop, up the ascending limb of the loop, and our distal convoluted tubule to the collecting duct. The collecting ducts converge and form the papillary ducts. And the glomerulus and this renal corpuscle produces a fluid. We're not calling it urine yet because we have a lot of work to do to clean that fluid up. So the fluid is going to be produced here, right? The glomerulus is fenestrated capillaries. What does that mean? What are fenestrated capillaries? They have holes in the walls, right? So bigger things can get pushed through. Still not the red cells, white cells, platelets, and large proteins. But everything else, glucose, calcium, magnesium, sodium, chloride, potassium, urea, all those things get pushed through those fenestrated capillaries into this capsule. And now we're going to call it filtrate or tubular fluid. And personally, I like filtrate, just makes more sense to me. Whatever works for you in terms of a terminology is fine with me, as long as you understand what it is that we're creating here. So the first thing is that capsular fluid. The filtrate is produced here in the glomerulus, and then it travels through the tubules, and here they are going to refer to it then as tubular fluid. Again, I think filtrate is fine. And you will see it referred to as filtrate in some of the references if you're reading up on the kidneys. This fluid, we know it's fluid, contains a lot of stuff. And that stuff is going to be recouped, it's going to be reabsorbed along the length of this proximal convoluted tubule. That tubular fluid is going to continue to be adjusted. Materials, electrolytes, are going to be pulled back into the blood via reabsorption. We have a special thing that happens along the loop here, and we'll talk about that when we get to it. And then the distal convoluted tubule, this right here, might see something referred to as secretion, where materials that didn't get pulled out of the blood or got put back in the blood, then get pulled out of the blood again, like potassium, gets kind of moved around. And remember, it's really important that we maintain a low calcium level outside our cells. We can have 3 to 5 milligrams per milliliter of potassium, or milliequivalent I should say, of potassium in our circulating fluids. We maintain a high concentration of potassium inside the cells. Why? Yes, keep going. Yes, all of our excitable cells are everywhere. All our excitable cells have to have that balance between sodium outside the cell and potassium inside the cell so that we can conduct action potentials, so that we can have depolarization, so we can get energy from one space to another. Good, so we're gonna keep an eye on that potassium because it's a key element in this whole process, especially the distal convoluted tubule. Once that fluid enters into our collecting ducts, then we can call it urine. So filtrate, up until that point, tubular fluid, up to that point, then we can call it urine, once we get it into the collecting ducts. Now, what we haven't mentioned so far is that these tubules are surrounded with capillaries, which is how we're going to get the material from the tubules back into the circulatory system. And those capillaries are referred to as peritubular capillaries. There's also, of course, around all of our tissues, in all of our body organs, tissues, there is a space between our cells. And that's no different between our tubules and the peritubular capillaries. So we have peritubular fluid that is essentially interstitial fluid between the structures. All right, so this is a closer look at our corpuscle, the renal corpuscle. One of the first things I want you to notice is that our afferent arteriole is much larger in diameter than the efferent arteriole. What's the benefit of that?
More nutrients and the bigger things can come in.
Yep, what else? Yep, plenty of room for blood to come into the glomerulus. What else? Yeah.
Would be more pressure?
Yes. Yep, pressure. So you are familiar with the garden hose. You put a nozzle on the garden hose. You can have full stream when the nozzle is wide open. You close that nozzle down, you build up pressure. That's exactly what's happening here. You have good big flow of blood into your glomerulus, and then you have a restriction of that flow out of the glomerulus, builds pressure in the glomerulus itself. And that pressure is going to be essential for literally pushing all of those solutes out of the blood and into the capsule. Pressure is important. We don't want too much pressure, right? Hypertension, no good, because that would increase the pressure too much. But we do have to have some pressure so that all of the materials that are in the blood get pushed out into the capsule and then ultimately into the proximal convoluted tubule, where we may be able to recoup them, reabsorb them, or not, depending on what our needs are. The capsule itself is made of simple squamous epithelial cells. And this is referred to as the parietal layer of the capsule. And then on top of each of these little capillaries, we have these specialized cells called podocytes. And they call them podocytes because of the way they interdigitate with each other. Right? Little toes interdigitating with each other. Podocytes. And you can see that here. And believe it or not, these create an even more important filtration process. They're called filtration slits. So you've got the fenestrated capillaries with the holes in them, and then the filtration slits are a little bit tighter. So things that might get past the fenestrated capillaries can't get past the filtration slits, like some of your medium-sized proteins. That filtration process creates the filtrate, and the filtrate is going to flow from the capsule to the proximal convoluted tubule. I learned the capsule as the Bauman's capsule. You will see it referred to as Bauman's capsule in some of the references, or you will see it referred to as the glomerular capsule. Either term is appropriate and fine with me. This is a histological slide image of the kidney. This is one glomerulus. Here's the capsule surrounding it, so collectively known as the corpuscle, right? There's another one, glomerulus, capsule, corpuscle, glomerulus, capsule, corpuscle. And it's showing you a lot of tubes. All these tubes are created by cuboidal epithelial cells. And believe it or not, in this image, I could tell you which tubes are proximal convoluted tubules and which are distal convoluted tubules, just by looking at the cuboidal epithelium and the lining of the tubule. So this is a distal convoluted tubule. This is a distal convoluted tubule. This is a distal convoluted tubule. This one, and this one, and this one, and many more are referred to as proximal convoluted tubules. And the reason I know they're proximal convoluted tubules is because the cuboidal epithelial cells that are forming those tubules are covered with microvilli. And the microvilli increase surface area. So we're able to reabsorb as much as we possibly can or need in the filtrate that's coursing through these tubules. So let's look at this again. Here's our glomerulus. There's the capsule. This is proximal convoluted tubule. Why do they call it proximal convoluted tubule?
Is it because of all the switchbacks?
Yeah, it's very convoluted.
Yeah, it's a
Yeah,
convoluted
it's
tube.
very convoluted.
Yeah.
Lots of twists and turns, packed in there. Packed in there. Can't even see your capillaries and your peritubular capillaries because there's so many tubules. But they're in there, they're around, they're tucked in between. You see a little, maybe some red cells right there, maybe something going on right there. Proximal, 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 proximal. Oh, distal.
So the distal doesn't have like the little.
That's right, it doesn't have as many microvilli.
And are there more sections of proximal convoluted tubes?
They are one
Tubules?
tube,
It looks like, yeah,
it's one tube.
of the tubule, there's more. It
Of-
looks like there's more.
Yes, there is way more of this than this.
Oh, yeah.
Way more of this than this.
Okay.
Because the majority of what's happening, the recouping, the reabsorption, the retrieving of the materials that we want to pull back into the system happens in the proximal convoluted tubule.
Okay. And the distal doesn't even have the microvilli.
Very little,
It has very little microvilli.
very little. Mhm.
Okay.
And in the distal, what we see more of is secretion. So
Oh.
materials that are still in the blood that we want to move from the blood to the filtrate are going to be secreted more often in the distal convoluted tubule.
So that's why you saw all the cuboidal really clearly, because there's more
Yep.
secretion happening there.
There's no microvilli.
And not
And
as
very
much
little
microvilli.
They call
Okay.
these proximal convoluted tubules stellate, from the word stellar, starry. That's what these, right? You can see it. Yeah, good. Distal convoluted tubule looks like maybe a collecting duct. You've got really nice cuboidal epithelium there in the collecting duct. Here's another distal convoluted, distal, proximal, proximal, proximal. Many more proximal sections. And I can't say these are all one tubule. You know, there's, again, 1 million nephrons packed into a structure the size of a bar of soap.
So each nephron has one tubule.
Each nephron has a proximal convoluted tubule,
In the
a descending limb, a loop, an ascending limb, and a distal convoluted tubule. Here's a little closer view of the glomerulus. This is your distal convoluted tubule, and then we're going to talk about why this is important here in a moment. But this distal convoluted tubule is tucked into what's known as the vascular pole of the glomerulus. Afferent arteriole bringing blood in, courses through our capillaries, and then out through the efferent arteriole, which in a better diagram would be smaller diameter. Remember, this has greater diameter, this is tighter, to increase the pressure inside the glomerulus so we can push all those materials out that need to be pushed out. We also have some specialized cells called mesangial cells. We'll learn more about them in a moment. These mesangial cells are important for helping maintain the blood pressure within the capillary bed, and they do some fairly major production of materials. So here you see it's called Bowman's capsule in this image. Once that blood has been filtered, the fluid that's collected in the capsule is sent off through the proximal convoluted tubule. So the proximal convoluted tubule here, as it says, the longest of the segments, lined with simple cuboidal epithelium with what's called the brush border. We know the brush border is formed by microvilli. Yep. And that gives those tubules that shaggy appearance, as it says. And this is where the vast majority of our reabsorption occurs, within the proximal convoluted tubule. Afferent in, efferent out, capsule, glomerulus are little podocytes covering the capillaries themselves. The fluid then that's produced and captured in the capsule is sent through the proximal convoluted tubule. These are representing your brush border, those microvilli. And then down through what's called the thick descending limb, and then there's a thin descending limb, a thin ascending limb, a thick ascending limb, distal convoluted tubule. And you can see, so they're representing some microvilli, but not as many microvilli on the distal cuboidal cells off into the collecting duct. And the collecting duct has some really interesting cells scattered along its length as well, as we'll find some of them in the distal convoluted tubule too. This portion right here is fascinating because this portion of the loop is impermeable to water. Water can be absorbed all along the length except for right here. This portion of the tube is permeable to sodium and chloride. And that sodium chloride is pulled out of the tubule, which then influences water in this segment and the movement of water. And it just said something wrong. This is permeable to water. This is not permeable to water.
Okay.
Yeah.
Just gonna ask that, like, wait a minute.
Yeah. Tired. So we have movement of water here, but not movement of water here. And when we pull sodium out here, it's going to pull water out here and put it all back into the circulation. What am I talking about? This is a good image because it gives you a better impression of where all the tubules are located relative to each other. So, cortex, medulla. Here's our arcuate artery to cortical radiant artery to afferent arteriole, glomerulus, afferent arteriole, capsule of the corpuscle. We have then our proximal convoluted tubule, descending limb, ascending limb of the loop, distal convoluted tubule. Notice that the distal convoluted tubule is tucked into what's called the vascular pole. And that's what I was trying to show you here. Vascular pole, this is distal convoluted tubule. And this too, distal convoluted tubule tucked into the vascular pole. This is important because there are specialized cells here known as the macula densa. And these cells are sensitive to sodium content. High sodium content means you probably have low water content. When you're thirsty, you have high sodium content, low water content. And the macula densa can then influence how the kidney deals with water concentration and sodium ion reabsorption. So it's always cool to notice that our macula densa is tucked into this vascular pole. And this is the best way to depict it. You can see that distal convoluted tubule tucked into this region. And essential because of the densa, macula densa that's located right there. So we've produced our filtrate, captured in the capsule, proximal convoluted tubule, where we have the filtrate coursing through. We're going to be reabsorbing materials all the way along the proximal convoluted tubule, down the descending limb, water reabsorbed here, not reabsorbed here, sodium chloride, potassium reabsorbed here, some reabsorption, some secretion here. This fluid then ends up in our collecting ducts. The collecting ducts end up in what are known as papillary ducts. And the papillary ducts drain into the pyramids of the kidney. And these are my favorite googly-eyed cells ever. Don't they look like SpongeBob SquarePants? No. So we have intercalated cells and we have principal cells. The principal cells respond to ADH. What's ADH?
the urine
anti-diuretic hormone. Antidiuretic
So you
hormone.
don't pee.
Antidiuretic hormone. Antidiuretic hormone encourages these principal cells, which are located along the collecting ducts and up into the distal convoluted tubule. These cells respond to ADH by creating what are called aquaporons. And the aquaporons are little proteins that then get embedded in the principal cell's plasma membrane and allow water to be pulled from the collecting duct and put back into the circulation. So we can recoup more water. If you are low on water, antidiuretic hormone is going to help you retain as much water as possible. So my antidiuretic hormone was working overtime yesterday. Because I'm telling you, I didn't drink nearly enough water. Thank you. Gabbing on. So these principal cells respond to ADH. These intercalated cells respond to changes in pH. And we have more than these two types, but we're going to just talk about the A cells and the B cells. The intercalated A cells respond to low pH, and they help to eliminate hydrogen ions, thus raising pH. And the B cells, the intercalated B cells, respond to a more basic pH and help to remove bicarbonate ions when our pH is too alkaline. So the kidneys are able to help us maintain our pH overall through these intercalated cells. These principal cells respond to ADH. Aldosterone, what do you remember about aldosterone?
Hormone.
It's a very important hormone. What'd you say, Ryan?
Sorry.
It is not a steroid
And then
hormone.
there's the.
I mean, it is protein hormone, but
It's the salt.
yes.
It was from the renal or the cortex of the
Adrenal,
the adrenal gland.
right? The
And there was the
zona
the
glomerulosa.
layers.
That sound familiar?
Yeah.
Zona glomerulosa and
And then
mineralocorticoids.
aldosterone.
Aldosterone, released from the adrenal glands, influences the kidneys to reabsorb sodium
Yeah.
along the ascending limb. What follows sodium? Chloride. What follows sodium chloride? Water. So we're going to retain water from the collecting ducts via principal cells. They're going to help us retain water from the ascending limb because they respond to aldosterone. Aldosterone stimulating these cuboidal epithelial cells to open themselves up to sodium and chloride and potassium at this point. And then later, we're going to get rid of those extra potassium through the distal convoluted tubule. So we're going to remember
By secretion, right?
by secretion.
Okay.
Yep. So we're going to remember more. We're going to see more, talk more about this as we go. This is just your first introduction to what's happening in the kidneys. Two types of nephrons in the kidneys. You got your cortical nephrons, and you have your juxtamedullary nephrons, close to the medulla. These juxtamedullary nephrons have one job only, and that primarily is conserving water, making sure we get as much water back into the system as possible, producing the most concentrated urine possible. Again, why do you drink a lot of water? So that you don't have that concentration, your juxtamedullary cells don't have to work as hard, and you urinate more often. These are about solute concentration, about our electrolytes, primarily for reabsorption and secretion, keeping a balance of those electrolytes, and water, and glucose, and all the other materials that are pulled out of the blood, got to be returned to the blood. So as I was saying to you earlier, our tubules are surrounded by these peritubular capillaries. So we're afferent in, efferent out, the efferent become peritubular capillary beds. These juxtamedullary nephrons have a specialized set of capillaries called the vasa recta. As you can see, it's really linear, and it's all about literally responding to a high concentration of urea in this medullary tissue. Urea acts as a sponge and pulls water, and then that water gets returned via these vasa recta capillary beds. So 15% of all of the nephrons in your body are juxtamedullary, the other 85% are cortical nephrons. And this is gonna round out our discussion. So this is just an overview of how the mammalian kidney works. And I didn't have a chance to pull it up, so just give me a chance to